All right. Hi again, everybody. Welcome back. I hope you managed to have a nice little break between this and our last lecture. So, this is going to be the third part of our lecture series, and this afternoon session is going to be on the development of the central nervous system. So, let's jump right in. Okay, so for this afternoon's lecture, what we're going to do is we're going to have a look at neurulation first, followed by the neural plate and what it is, before we move on to the development of the spinal cord, the brain vesicles, as well as. Okay, so just before we start looking at neurulation, let me just preface by saying this. So the nervous system consists of two main structural divisions, right? You have the central nervous system and you have the peripheral nervous system. The central nervous system itself consists of the brain and the spinal cord, and the peripheral nervous system consists of the rest of the nervous system that is outside of the central nervous system. So that means things like excuse me, the spinal nerves and ganglia, the autonomic nerves and ganglia, as well as the enteric nervous system. So, for the purposes of today's lecture, we're only going to be looking at the development of the central nervous system. All right? Okay, so let's begin by going back to the bilaminar disc. Let's see if you remember the bilaminar disc. So if we look at image one up here, you notice these two layers, right? You have the epiblast layer and the hypoblast layer. So at this stage of development, with a little bit of cell signaling, a little thickening occurs on the surface of the epiblast layer, and this leads to the formation of this primitive streak, and kind of towards the cranial end, you have another structure beginning to form, which is the primitive node, and we'll get into why these two structures are important. And so as development progresses through the next few days, so this is if you kind of, the image number two down here is basically these two images kind of turned around, right? So image one kind of on the left, and the right is a turned around, rotated clockwise version of image one. So looking down here, same thing at image two. This is what I was talking about, where the cells of the epiblast layer, kind of a thickening occurs on their surface, the and that primitive streak forms. So what happens is that some of the cells, at the centers of both this primitive streak, well here, the primitive streak and the primitive node, they start to apoptose, or they start to die. And this apoptosis of these cells basically kind of creates a sort of cavity where there used to be cells that have now apoptosed, right? So in turn, this cavity now allows for the movement, or rather the migration of cells through this cavity to be repositioned and to kind of settle down in a new place. So before apoptosis, we have the primitive streak, and when apoptosis occurs, that primitive streak now becomes a primitive groove. So same thing likewise is happening at the primitive node. Before apoptosis, you had a primitive node, and when apoptosis occurs, it becomes a primitive pit. Okay, so what is neurulation then? Neurulation is basically the formation of a neural tube from this sort of flat layer of ectoderm. And that's the basis of it. But in order for the neural tube to actually form, something else has to happen first. So the notochord has to form. So let's explore how the notochord forms. Okay, let's remember again that epiblast layer of cells, that are getting ready to migrate down to form a trilaminar disc from being a bilaminar disc. So you had the bilaminar disc, which was the epiblast cells and the hypoblast cells, and then now that's going to become a trilaminar disc that is composed of endoderm, mesoderm, and ectoderm, right? Now, while a lot of these cells migrated down the primitive groove to form those three germ layers of endoderm, mesoderm, and ectoderm, some of those cells end up forming a sort of kind of like a collection of cells somewhere in the midline of the embryo. So what happens is at the primitive node sort of extends as a sort of tube of mesenchymal cells that exists in between the ectoderm and the endoderm layer, right? Um, and that is, in fact, the notochordal process. So, in those early days of development, the notochordal process is going to continue growing in kind of like a cranial direction, and by the time we get to, say, the 20th day of development, what happens is that the notochordal process will start to fuse with the endoderm that is right under it. So that's what you're seeing in this image here. You have the notochordal process up here kind of fusing with the endoderm layer right under it to form the notochordal plate. And following the formation of the notochordal plate, eventually the cells of that notochordal plate are going to kind of lift off. So they kind of fuse to form this notochordal plate, and then the cells of that notochordal plate they lift off and they separate from the endoderm layer, and they sort of kind of pinch off of the endoderm layer and form basically what kind of resembles a sort of rod that basically extends the entire length of the embryo, and this rod itself is the notochord itself, basically. All right, so that is the process of the formation of the notochord. What about the neural plate? Let's start to put these pieces together to see what's happening. Okay, so before I describe these 5 images to you over here, I'll just say this. So imagine the notochord as sort of air traffic control, where, air traffic control tells planes which runway they can land on, which runway they can take off from. It controls the traffic of the planes in the sky. Basically, the notochord does the exact same thing. So it's really the signaling center that tells the cells of the ectoderm what they should do. So at this stage that we're in development, as the notochord is forming, the ectoderm in the middle of the embryo also kind of starts to thicken and starts to become what is known as a neural plate. And the ectoderm kind of starts to become what is known as neuroectoderm. Basically, the neural plate will span the entire craniocaudal length of the developing embryo, where at the cranial end it's going to appear a little bit wider and eventually become the brain. And kind of the rest of the neural plate will basically give rise to the spinal cord. Um, okay, never mind. Let's just look at these 5 images here. Right, let's orientate ourselves. So in these series of images, you're going to notice that in blue over here, we have the ectoderm layer. We have the mesoderm layer here in red, and you'll notice as these images kind of progress, the red layer starts to look a little bit different, and that's just basically the mesoderm differentiating, into its different parts like paraxial mesoderm, lateral plate mesoderm, and so on. And in yellow, you have the endoderm layer, and finally in black in the middle here, you have the notochord. Okay, so we start off over here looking at the blue in image 1. We have the neural plate. So you'll notice as we get from image 1 to image 2, there's a sort of kink that appears, a little bend that appears in this neural plate. So the lateral edges or kind of the sides of the neural plate start to become elevated a little bit because they're beginning to fold. And in this process, where the sides are kind of rising a little bit, beginning to fold a little bit, they create this little area known as the neural groove, which, as you can see, as the images progress, that neural groove gets deeper and deeper. Now, as all of this is happening and these lateral edges are kind of rising, they of course become narrower and longer. The lateral edges become narrower and longer, and excuse me, and these raised edges of the neural plate look very different if you compare images 1 and 3, and you can see that it's a lot more raised. Obviously, where you start at flat, it's a lot more raised in image 3. So we start calling these raised lateral edges the neural folds. Now, by the time you progress to image 4, you'll notice these little black specks that have appeared in the image. These top parts of the neural folds that will eventually come together are parts of the neuroectoderm that are brought towards one another to meet. These are the neural crests, so these little black specks. Now, eventually, the two sides of the neural plate, they come together, they meet, they fuse, and they form this sort of tube of neuroectoderm that's going to run down the length of the embryo. And it's basically open on both ends, and it's just a tube that runs down the length of the embryo. And that's basically what you see here in image 5, is that neural tube running down the length of the embryo. This neural tube will eventually, pinch away from the ectoderm. So it pinches away from the ectoderm, and the ectoderm itself kind of reforms over the neural tube. And later on, as development progresses, it will form the external portion of the embryo. So looking at the neural tube, it itself is termed neuroectoderm, and it's going to run the length of the embryo. And this neural tube is basically what is the precursor to the central nervous system. So, as I said a minute ago, the neural tube is going to stretch cranially and caudally, and both ends of it are kind of left open at the cranial and caudal neuropores. The cranial neuropores are going to close somewhere, around the third week of development, or around the 24th week of development. And the caudal neuropores, and neuropore is spelled Neuropores. The caudal neuropores close a bit later, somewhere around the 26th day of development. And basically, with that, neurulation is complete, and the neural tube, has been fully formed by the end of the 4th week of development, with its anterior region being slightly larger and ultimately forming the brain, and its posterior region being, slightly more tapered, slightly narrower, and ultimately forming the spinal cord. And remember these little black specks we spoke about on the crests of the neural folds that come away or detach and form a new cell population? Those were the neural crest cells. And as these neural crest cells break contact with the neural tube, as you can see here in image 5, they're basically able to, leave and carry out their different fates, and, contribute to whatever derivatives it is they need to contribute to. Now, some of these neural crest cells will form much of the peripheral nervous system, but the rest of these neural crest cells are going to have various other fates in the. So this is just a sort of little illustration of what I just described, with the neural folds forming. Let me get my pointer out. The neural folds kind of forming up top, giving rise to the neural tube here. So the ectoderm layer is actually responsible for a few things. It's going to help the process of neurulation along. It's ultimately the presence of the notochord allows for the formation and the shaping of the neural plate that's going to give rise to the neural groove, later on the neural folds before they pinch off to form, the central nervous system and later development. So I hope this kind of helps you. It kind of helps. Okay, so let's take a minute to discuss the clinical considerations if anything goes wrong during these little parts of development that we just discussed. So, the most common congenital abnormalities of neurulation are going to be associated with neural tube defects. So, remember how I said the neuropores at the cranial and caudal ends of the neural tube are the last to close? So, defects are most likely to occur at those cranial or caudal ends. Now, if the neural tube fails to close caudally, it's going to affect the spinal cord as well as the tissues that overlie it. And this includes the meninges, the vertebral bones, the muscles, and of course, even up until, the skin. So what we're looking at here is spina bifida, which is a condition in which the vertebra fails to form completely. And of course, there are various degrees of severity to this. So, looking at most severe over here um, to least severe over here. So, you have spina bifida occulta, which is the least severe form, with just like maybe a tiny gap forming in one or more vertebra in the region of the level of L5 to S1, and this will cause very little or actually sometimes even no symptoms at all. Instead, a patient might present with this little unusual tuft of hair that might be present in that particular region of the back instead. The next type is slightly more elevated, is a slightly increased severity of spina bifida, which is spina bifida meningocele, and it's the failure of the vertebra to fuse, and this region tends to often be large enough to where it actually allows the meninges of the spinal cord to sort of protrude externally. And of course, in this rightmost image, you have a sort of more elevated and more severe case. If the spinal cord or the nerve roots also protrude, here you have spina bifida with meningomyelocele, and this condition might affect things like sensory or motor innervation at the level of the lesion. And of course, it might potentially affect other organs as well, like the bladder, like I said, depending on the level, the vertebral level at which the lesion has occurred. So, some other examples of clinical considerations in the event that the neural tube does not close are things like exencephaly or anencephaly. And in the event that the neural tube um, may fail to close at its more cranial end, it'll of course cause abnormal brain and calvarial bone development. And of course, in some instances, some of these anomalies might include the brain possibly being partly or entirely outside the skull, as is in the case on the left here, which is exencephaly, or in other instances, like on the image on the right here, the forebrain might fail to develop entirely, and that is known as anencephaly. Now, exencephaly might precede anencephaly, and exencephaly is actually, in some cases, it might be an early stage of anencephaly, and if a pregnancy actually proceeds with an exencephalic embryo, the neural tissue eventually will degenerate, and unfortunately, for an infant that's born with exencephaly, they have a rather poor prognosis. Whereas on the other hand, with anencephaly, major parts of the brain or the skull or even up to the scalp may be missing. And whilst the etymology of the word anencephaly itself might suggest the absence of a brain entirely. Instead, what it is that some infants are actually born without the telencephalon region, which, if you can think back to your other lectures, would actually, the telencephalon region is actually responsible for many functions, and of course, everything else like that overlies it, like the skin, the bone, the meninges, of course, would not be present as well. And ultimately, anencephaly is also not consistent with life, and infants with this order do not survive. Okay, so following the process of neurulation, the central nervous system is going to continue to develop at the cranial end. As the cranial end of the neural tube starts to dilate, it starts to fold and starts to form all the spaces that will eventually become, the shape of the brain that we're used to identifying it as. And of course, like I mentioned earlier, the remainder of the spinal cord will become, the remainder of the neural tube will become the spinal cord. So, as we mentioned a little earlier, the central nervous system is going to be made up of the brain as well as the spinal cord. So, let's start by looking at the spinal cord. So, on the caudal end of the neural tube, it's going to continue to elongate enough to form the spinal cord, right? So, let's just briefly discuss the fact that the spinal cord differs a little bit in a developing embryo versus how it presents in adults. Now, in an embryo, the spinal cord reaches to the very caudal end of the vertebral column, and the closer we get to the end of the embryonic period, which, if you remember, was week 8 of development, what's going to happen is that the vertebral column itself is going to start to grow quite rapidly. And in growing, as the vertebral column grows, it basically causes the sort of tail region or the end region of the spinal cord to reach the level of sort of L3 vertebra by the time of birth. So the vertebral column ends up being longer than the spinal cord itself. And then from birth through to adulthood, the terminal portion of the spinal cord will, of course, continue to rise, so that technically, by the time we get to the 24th week of development, the spinal cord basically stops at a level of the S1 vertebra, which means that the end of the cord really rests around kind of level of L2 or about L3 in a newborn. But in adults, the spinal cord technically stops at a level of the L1 vertebra. So because there is this difference in length, the lumbar and sacral spinal roots are going to sort of project kind of in an oblique manner from the spinal cord to their vertebral levels. And eventually, of course, the nerve roots at the end of the spinal cord will form the conus medullaris, with the nerves kind of branching out more inferiorly still to form the cauda equina. And in terms of things like the subarachnoid space, the arachnoid matter, and the dura mater, all of these are going to extend to the level of S2. So that's for the spinal cord. So let's try to zoom in a little bit, take a slightly closer look at the spinal cord now. Okay, so the spinal cord, like we said, develops from the caudal region of the neural tube, and the caudal eminence. And in fact, what's actually happening is that the neural tube region that is caudal to the fourth pair of somites are going to start to develop into the spinal cord. So in this region of the neural tube, its lateral walls will actually start to thicken, and because its lateral walls are thickening, what happens is that it makes the neural canal smaller because its lateral walls are thickening, right? So by the time you get to the 9th week of development, what you basically have is a lumen in the center of the spinal cord known as the neurocele, where you can see in this image up here on the top right hand corner, or the neural canal, which will later on become the central canal. Now, this neural canal is lined with layers of things like neuroepithelia, or otherwise basically known as the ventricular zone. Now, the cells of this ventricular zone are going to start differentiating into various things like primordial neurons called neuroblasts and glioblasts. So to differentiate these two, the glioblasts are basically going to support, they're going to form supporting cells or neuroglial cells, and the neuroblasts in turn will become neurons and migrate out to form the mantle and marginal layers you see down here. And really, just to simplify it for you briefly, the mantle layer of cell bodies will then form the gray matter, while the marginal layer will sort of surround this layer, and it contains the nerve fibers which arise from the neuroblast cells of the mantle layer. And anyways, these nerve fibers ultimately become myelinated, which in turn gives rise to this sort of white colored appearance. So this part, or rather the marginal layer, ultimately becomes the white matter of the spinal cord. So the mantle layer is the gray matter, and the marginal layer becomes the white matter. So you don't really need to get into the technicalities of the explanation on this part. But it is worthwhile to note here that from roughly around day 20 of development to about day 35 of development, the meninges will start to develop from cells of the neural crest and mesenchyme, and these cells will migrate and start to surround the neural tube to kind of form basically the primordial meninges, with the outer layer ultimately thickening to form the dura mater, and the inner layers forming the pia mater and the arachnoid mater. And of course, eventually, as development progresses further and further and further, by the time you get to the 5th week of development, cerebrospinal fluid will also slowly start to form. So that's just a brief rundown of what is happening with the spinal cord. Okay, then, what about the brain vesicles? What's happening with the development of the brain vesicles? So the brain starts to develop quite early on, actually, roughly around the 3rd week of development, when the neural plate and the neural tube are forming from the neuroectoderm. So when the neural folds and the cranial region start to, fuse, and the more rostral neuropores start to close, what forms is 3 brain vesicles. So these 3 primary brain vesicles develop in the cranial end of the neural tube as the prosencephalon, or otherwise known as the forebrain. You have the mesencephalon, or otherwise known as the midbrain, and the rhombencephalon, or otherwise known as the hindbrain. So now, as these 3 primary brain vesicles start to grow, by the time we get to week 5 of development, they've developed even more, and eventually these 3 primary brain vesicles become 5 secondary brain vesicles. And how this happens is basically that the region of the forebrain, so the prosencephalon, and the region of the hindbrain, so the rhombencephalon, basically split. So in the forebrain, or the prosencephalon, it splits to form the telencephalon and the diencephalon, while the region of the hindbrain or the rhombencephalon splits to form the metencephalon and the myelencephalon. So you go from 3 primary brain vesicles to 5 secondary brain vesicles. And of course, around the same time, the brain is rapidly growing, and as it grows, it ends up kind of bending a little bit ventrally in line with how the head folds, with how the head is growing, which ultimately produces that sort of midbrain flexure in the region of the midbrain, and you have another flexure in you have a cervical flexure at the junction of the hindbrain and the spinal cord. Okay, so you've got all these different regions of the brain. What actually happens to all of them? So very broadly speaking then, this is what happens to all of those different parts of the brain. Basically, the dorsal region of the telencephalon of the forebrain is going to form the cerebral cortex, while its more ventral region will form the basal ganglia. So that's for the telencephalon. With the diencephalon, what's going to happen is that it's going to form these structures. It's going to form the optic cup and stalk, the pituitary gland, the hypothalamus, the thalamus. This shouldn't be thymus, this should be thalamus, if you could just please make that correction, as well as the pineal body. So that's for the diencephalon. The myelencephalon of the brain becomes the medulla oblongata, while the metencephalon regions becomes ventrally it develops into the pons, and dorsally it develops into the cerebellum, and lastly, this the mesencephalon, of course, we said is the midbrain. So the midbrain, the pons, and the medulla oblongata together form the brainstem, as you know. Okay, then moving on to the ventricular system. Okay, so how then does the ventricular system actually develop? So remember we had a neural tube, right? Well, the region of the lumen of the neural tube, that's basically where the ventricles form. And these ventricles contain cerebrospinal fluid, or CSF, that is produced by the choroid plexus. So the cavity of the diencephalon here is known as the third ventricle, and it forms from the cavities within the telencephalon and the diencephalon via the ventricular foramina of Monro, and it's connected on both these sides to each of the lateral ventricles. All right, so this is really just a very simple, plain cartoon schematic to kind of give you an idea of like the flow of these ventricles. All right. So looking at the mesencephalon, the ventricular system should be a little bit narrower here, and this narrowing is actually called the aqueduct of Sylvius, or just more simply put, the cerebral aqueduct. And this pathway really just allows the cerebrospinal fluid to flow between what is up here known as the third ventricle and down here known as the fourth ventricles. And of course, if you have any sort of obstruction to the flow of the cerebrospinal fluid, here it can cause the condition known as hydrocephalus, which we'll talk about in a moment. And finally, the cavity of the rhombencephalon, which is also continuous with the central canal, is also known as the fourth ventricle. So basically, to summarize what you see in this image here, the lateral ventricles of the telencephalon kind of link with the third ventricle over here of the diencephalon, which is going to be connected via this cerebral aqueduct of the mesencephalon to the fourth ventricle of the myelencephalon, which later on just kind of continues as the central canal of the spinal cord. Okay, so what then are some clinical considerations of developmental anomalies that can occur during this part of development? Okay, so looking at this image here of portraying an encephalocele, this occurs when there is a defect in the cranium, which then results in the herniation of the intracranial contents through that defect that's been created. And this herniation, of course, can have varying levels of severity. So it might just include, a herniation of the meninges, or it could be a herniation of the meninges and maybe even parts of the brain, or in more severe instances, it could include herniation of the meninges as well as parts of the brain, as well as parts of the ventricular system. This condition, of course, depending on its severity, might actually be repaired. Moving on to this one, the condition depicted here is hydrocephalus, which is basically a condition that results from an excess of cerebrospinal fluid within the brain. And hydrocephalus can basically result from a blockage of flow or a failure to resorb cerebrospinal fluid, or even in instances where there is an increased production of cerebrospinal fluid. And one of the more common causes of congenital hydrocephalus is because of congenital aqueductal stenosis. So there is that the passage between the third and fourth ventricles in the brain is either blocked or there's stenosis, so it's too narrow to allow for the cerebrospinal fluid to be able to adequately drain. And ultimately, this fluid accumulation will eventually cause hydrocephalus, which in turn causes the sort of increased pressure to be built up inside the skull, which in infants causes a rather quick increase in head size. Hydrocephalus can also be treated, and treatment options include things like possibly inserting a shunt system to try to help to open that stenosis or that blockage, or in some instances, even being able to redirect the cerebrospinal fluid to an area where it can be reabsorbed instead. Okay, so that was today's lecture on the development of the central nervous system. I know today was a longer one because we had two back-to-back lectures. So thank you for your time. I hope it was fruitful for you. If you have any questions, please, again, as I have mentioned in my earlier lectures, feel free to email me directly at antskr@nus.edu.sg. If you prefer to reach out more anonymously with your question, you can hop onto that bit.ly link at What's Your Anatomy Question. Just drop me a little line. I will ultimately compile questions and send them through to your class rep, who can then disseminate it to the rest of you. I hope you have a good rest of the afternoon, and I will see you in a couple of weeks for the last segment of this lecture series, where we look at the development of the tongue and palate. All right, have a good one, everyone. Goodbye.